Hey everyone, it's Steven again, and this is my next video for the Cowan Microbiology textbook, and these are the slides that were created by my class at Sinclair Community College. So these are not my slides. The point of me making these videos is just so that I could get a good review for the test we have on Tuesday, and hopefully these reviews benefit someone out there. I'm not the most non-monotonous person. But we'll do the best I can, and I'll go through these real quick. These are only 25 slides for this lesson, and these are the basics of biochemistry, essentially. You would have learned most of these in any general chemistry course you've had, along with any general biology course, so it's basically biochemistry. So here we go. So these are the things we're going to go over, some of the atoms and elements, the difference between a molecule and a compound, the different types of bonds that are important in microbiology, acids and bases, the basics, and some functional groups of molecules and the types of macromolecules. Okay, so all matter is made up of individual building blocks we call atoms. So these are basically the smallest particles you can find. Of course, you can go subatomic, obviously, but that's we're not going to go there here. It's defined as a tiny particle that cannot be subdivided into smaller substances without losing its properties. So basic parts of an atom, you got your protons, which have a positive charge, neutrons, which have no charge, and electrons, which have a negative charge. And I'm sure you guys have all heard the joke about the neutron walking into a bar, and then the bartender saying, "For you, no charge." So that's what they're talking about there. Basically, each specific combination of protons and electrons makes up an element. So if you look at a periodic table, you'll see that all the different elements have different numbers of protons, and that's what gives them their atomic number, essentially. And here we can see the basic structure of an atom. We have a nucleus at the center, which is composed of the protons and the neutrons. And then we have different electrons that are surrounding them in orbitals like this. And the shells look different depending on the hybridization, but we won't go into that for this course. Don't need to know that here. So as an example, the carbon atom has 6 protons and 6 neutrons, and it has 6 electrons that orbit it, but the protons and the neutrons make up the nucleus. Okay, so elements rarely exist in a pure uncombined form, meaning that things are usually combined together. When they're bound together, they're called molecules. This is what we refer to as a combination of 2 or more atoms. So if you have something like sodium chloride, you have a sodium atom bound to a chloride atom. The difference between a molecule and a compound is that a compound is a molecule made up of 2 or more different elements. So basically, for a molecule, the atoms don't necessarily have to be different. We can have we can have what's called a diatomic molecule, like oxygen, O2, or nitrogen, N2, and that's how they're frequently found in the air that we breathe, because they don't typically float around by themselves. When a molecule is formed, the atoms lose their individual characteristics and take on the properties of the combined substances. And the molecules are held together by bonds, which we will go over in a second. So there's 3 types of bonds that are important for chemistry. We have covalent bonds, which are where the electrons are shared between atoms. So we have, it's not shown here, but you'll see a picture eventually of 2 atoms that are next to one another, and they share an electron between the 2 of them in the orbital. We have what's referred to as an ionic bond, which is where electrons are transferred between atoms, creating charged molecules. So for the sodium chloride example, the sodium would usually have a positive charge, and the chloride would have a negative charge. Excuse me. And then we have hydrogen bonds, which are they're less bonds between atoms than they are between molecules. It's what you have between your water molecules. So you have your H2O, so your O oxygen. Your hydrogen atom, will be bound to another oxygen atom of a different water molecule. And these are attractive forces between those two called hydrogen bonds. It doesn't necessarily have to be oxygen, it could be nitrogen as well. So it's usually atoms that are very electronegative or atoms that want to hog electrons to themselves. And here's a visual on that. So we have covalent bonds. You can see we have an electron from either atom being shared between the two to make the molecule there. If we're sharing 2 electrons, we can do that as well. And you can see there's a total of 4 electrons there in that middle shared portion. For an ionic bond, we have, so let's say this is a sodium atom, because sodiums have 1 proton, 1 electron, as you can see from the periodic table, and it's donating 1 of its electrons to make that bond between the sodium and chloride there. This is just an example, but that's what it would look like. And then for a hydrogen bond, you have your electronegative molecule here. This could be oxygen or nitrogen, for instance. It's bonded to a hydrogen via a covalent bond, like this guy. And those hydrogen bonds are connected between those. It's called a dipole moment, meaning that this hydrogen takes on a partial positive charge because because the atom over here is a negative charge, so it's pulling electrons this way, so it's making this more of a positive charge. And this other negative atom over here, part of a different molecule, is attracting those two. So just to go even into more detail, covalent bonds, as you can see, we have 2 hydrogen atoms that come together to form H2 by sharing both of their electrons together. As you can see, the old saying, 2 heads is better than 1, or 2 hydrogens is better than 1. Molecular oxygen does the same thing. It's another diatomic molecule where it shares these 4 electrons here. Methane is a gas, and that's carbon and 4 hydrogens there that are covalently bound, like so. Yeah. Okay. Covalent bonds and polarity. I kind of touched on this with the hydrogen bonding, but basically when you have a very electronegative element like oxygen, it tends to want to hog electrons to itself and gives it more of a negative charge character. And this in turn gives those hydrogens that are involved more of a positive charge. Ionic bonds. Again, we talked about sodium chloride. So you have a sodium atom here where it's got 1 electron in its outer orbital, donating that to chlorine to its 7 electron orbital to make it a full 8. And if you remember from your general chemistry class, you want to have a full 8 electron orbital in the final shell to make it a stable molecule. You either want 8 or you want 0. So this way both sides get what they want. Sodium gets its 0 electrons here on its outer orbital, and chlorine gets its 8 atoms, so they're both more stable as a result, and so they bond together like so. That being said, though, if you put them in a glass of water, for instance, like a sodium chloride molecule in a glass of water, it tends to separate into its substituent charges. So you have free sodium and free chloride here because they bind to the different water molecules. So you have chloride binding to that partial positive hydrogen there and here as well. And you have the partial negative character of the oxygen binding to the positive sodium charges here. Hydrogen bonds, again, they're more between different molecules, like water molecules are the big example here. The negative oxygen binding to the positive hydrogen. They happen because of covalent polarity. Polarity basically just means the difference in charge between the oxygen and hydrogen, making this a negative and this a positive. They're weaker bonds than, say, a covalent bond would be between the oxygen and the hydrogen as part of this molecule. So this bond is going to be weaker than this bond. Acids and bases. So here we have, whenever you have a solution of water, you're going to have some portion of those water molecules that are dissociating into hydroxide ions, or hydrogen ions here for simplicity, and hydroxide ions. And a solution is acidic when there is an excess of H plus ions in solution. Excuse me. In reality, these tend to be H3O pluses rather than H pluses, but for simplicity, they say H pluses. But a solution is acidic when there's excess H plus ions in solution. A solution is basic or alkaline when an excess OH minus ions are in solution, like so. Water is neutral because it has equal numbers of H plus and OH minus ions, so it tends to be a pH of 7, which means it's neutral. And the pH scale is a measurement of the concentration of H plus and OH minus. And here's some of the different substances that are on that pH scale, with more acidic substances being over here, more basic substances being over here. Basic, another term is alkaline. Biological molecules. So organic molecules contain both carbon and hydrogen. This is what we refer to when we say organic. So like organic chemistry, that's the type of things you're usually going to be working with in that lab. These molecules contain special arrangements of atoms called functional groups, and these functional groups give those molecules a different character, different properties, depending on what they are. They can be, an example is a hydroxyl group, so if it's got an extra OH group on there, it tends to want to bond with water more. Another functional group could be a ester, which is a double bond with a, well, I guess I can't really talk about it without drawing it, so I won't explain it too much, but you'll deal a lot with those in organic chemistry if you take that class. Organic molecules come together to form macromolecules, large compounds assembled from small molecular subunits. Not sure if there's more to say on that. So these are some of the different functional groups that I was trying to talk about earlier. There's a hydroxyl group, for instance, with a OH group. A carboxyl, meaning it's got an oxygen and then another hydroxyl group, and then so on. Amino acids, so, excuse me, not amino acids, just amino group, NH2, and sulfhydryl, SH. And you can see the different examples there. There's 4 families of macromolecules, which if you've ever done, besides just chemistry, if you've ever done like nutrition, you'll know that there's carbs, lipids, proteins, and nucleic acids. We don't really consume these per se, but they're included in this group as well. Carbohydrates, we have general sugars or saccharides, and with the general structure being a carbons and waters, because it's carbon and hydrates. Lipids tend to be long carbon-hydrogen chains that are not soluble in water. So these are the things that are not so good for us. Proteins, amino acids combine in a polypeptide chain, which fold into specific shapes. And these basically give us our body's function, essentially. All of our enzymes, a lot of the items that give our cells the ability to do what they do, to move around, to chop things up, to break things down, has to do with proteins. So these are very important for biochemistry. So here's what the carbohydrate looks like. It can either be in different forms. It can be a single sugar by itself, which we call a monosaccharide, like this, like glucose, for instance. It can be a disaccharide, which is where you have 2 sugars bound together, like so. An example is lactose, which you have a combination of glucose and galactose bound together right here, or we can have a chain of sugars like we have right here. An example here would be the glycogen that's stored in your muscles. So if your body's in like a fight or flight mode, if you need to escape from a bear that's charging towards you, you'd use that glycogen to as a store of energy to get away quickly. Now these are not typically sustainable energy sources. They're more like, if you need them, in case you need them, you can use them real quick and go. The lipids tend to be more the long-term energy sources, as we'll get into in a second. So here's an example of, here's what glucose looks like. You have the Fisher projection and you have the, gosh, I can't remember the name of these types of drawings, but I think this is a Fisher projection and this is, I don't know. But we have an aldehyde group at the top of these. Here's another monosaccharide, galactose. So these two combined together would make up lactose, which is the sugar in your milk. It's a disaccharide. And fructose, which is mostly in fruit, has a ketone group up here. And it's also, as you can see, although it's still got 6 carbons like a glucose or a galactose would, it's arranged in a 5-carbon form known as a pyranose. Here's what lipids look like. So you have a glycerol molecule here attached to 3 triglyceride tails, and there's different types of these depending on if it's unsaturated or if it's saturated. If it's saturated, these tend to be all uniform carbons and hydrogens all down the line. If it's a unsaturated fat, you'll see like what are known as kinks in this chain or double bonds there. And so what happens is they don't stack as well together if they're unsaturated. And then they combine, this glycerol and these fatty acids combine to form a triglyceride. So you have a glycerol molecule up here and you have the fatty acids combined via ester bonds, like so. And these are more for energy storage as opposed to quick energy like our glycogen would be. Phospholipids are also important types of lipids. These are the lipids that make up our cellular membranes, as mentioned here. And what these look like is you have a phosphate group up here that's mostly a negative charge. These tend to be very hydrophilic. These tend to want to bind with water because of their charge. So kind of a quick hint is if something's charged, it tends to want to bind with water. It tends to dissolve in water very well, as opposed to the fatty acid portion here, which is very hydrophobic because of all of its carbons and hydrogens. Those don't dissolve well in water. And so what you'll have is the water surrounds, like this is the outer part of the cellular membrane. The water wants to surround the outside. And here you can see what this looks like. If you were to put a bunch of these in water, this is what would happen. They would congregate like this. This is called a micelle. So you have all these different phospholipid, all the tails want to avoid the water, so they're on the inside, and then all the phosphates form like this. This is what a phospholipid bilayer would look like in your cellular membranes, which is where you have water on either side of them. And you have the phosphate groups on the outside and then the tails on the inside. Here's another picture showing that. Within the phospholipid membrane, we have sterols, steroids, and the most famous one is cholesterol. These help keep our cellular membranes more stable. Proteins are the building blocks of life. So they're made up of 20 different amino acids. These are a few examples of those. All of the different amino acids have different characters. Some of them are hydrophobic, some of them are hydrophilic, some of them are negative charge, some of them are positive charge. And we typically need all of those some of them we need in our diet. Those are called essential amino acids, and then non-essential amino acids are amino acids that we don't need to consume, we can make on our own. There's different structures that go along with proteins and that are important for knowing sort of their function, because as they say, form follows function. Or wait, function, I can't remember which comes first, the chicken or the egg. But basically, for the primary structure of proteins, you look at the different order of the amino acids, so like this, up here. The secondary structure, these amino acids start to coil a little bit in different forms. We have an alpha helix here, and you can see those little red portions as their interactions. Or we can have beta sheets, like so, and these are the two main secondary structures involved in proteins. For tertiary structure, what happens is these sort of fold on themselves again, and we can have interactions like a disulfide bridge, which is where you have a sulfur on each portion connecting each other. And then if we have different so say this is like a protein by itself usually as a tertiary structure. If we combine different proteins together to make up one bigger protein, we usually refer to that as a quaternary structure. And they're usually showed by these different ribbon diagrams to make them easier to see. Nucleic acids are important for our DNA. They make up our DNA. We have two different sugars that make up the backbone of that DNA. We can have deoxyribose making up DNA, or ribose, which makes up RNA. And the big difference between these two is, well, what's this called? Deoxyribose and ribose. So we're looking at this carbon, we look at this oxygen here. See, we have an oxygen OH group on the ribose, but not on the deoxyribose. And this has an important impact on the structure of that, an important impact on the structure of the RNA or DNA, respectively. Basically, this inhibits the different sugars from stacking onto one another as well. So it tends to be, an RNA molecule tends to be less stable for this reason, because it has this oxygen group here. Another important distinction between these two is that these DNA and RNA both have different nitrogenous bases that go along with them. So both of these two have adenine, guanine, thymine, and cytosine. Both of them have adenine, guanine, and cytosine, but DNA has thymine and RNA has uracil. So these are the two distinctions here. And the purine bases are adenine and guanine. That's what this general structure is called, where you have two sort of circular molecules that are connected to one another here. And the pyrimidine bases are these guys, the thymine, cytosine, and uracil. Here's the general structure of DNA and RNA. This is what the backbone looks like. You have your sugar here, so your 5-carbon sugar, and your phosphate group. And then down this line we have our different nitrogenous bases. That's typically an RNA, because RNAs are typically singular-stranded. DNA are typically double-stranded, so you'll have two strands that are interacting with one another through their bases. If it's so we have cytosine that goes with guanine and adenine that goes with thymine over here. So here we got an A group, so that's got to be a thymine, and these aren't labeled, but you would see a guanine and cytosine there. And typically those guanine and cytosine bonds are stronger than the adenine and thymine bonds, because there's 3 hydrogen bonds that are associated with those two, and that's not really shown here, but that's just kind of extra information. One second. As I mentioned before, DNA is a double strand, so it forms a double helix when it is in its hot forms. This is just another depiction. So we have a adenosine here, which means it's combined. It's the sugar and the nitrogenous base. It can be an ADP, which means it has 2 phosphate groups, or it can be an ATP, which has 3 phosphate groups. These are some of the different types of carbohydrates, lipids, proteins, and nucleic acids for your reference, and what they're sort of important for. All right, hope you enjoyed. If you have any questions, please let me know. Thanks.